Hi, my name is Kimberly Kaus, and today I'll be completing the biochemical testing for microbial identification laboratory assignment. Today is January 30th, 2018. In advance, I have prepared my clean work surface by sanitizing with a 10% bleach solution. I have labeled the two vials labeled MRVP broth. One is labeled E. coli, one is labeled S. epidermidis. That'll be used at a later time, and they're both room temperature. I'm going to start. So I'll begin the experiment by opening the MRVP broth labeled E. coli. Fan it over the open flame. Open the nutrient broth with E. coli. Fan it over the flame. I am going to apply 4 drops from the E. coli broth into the MRVP broth, I believe. 3, 4.
Just drop it in a sec.
Fan them over the flame before. I'm fanning the MRVP broth labeled E. coli before capping. We'll store that for 48 hours. And we're going to repeat the process with the S. epidermidis. And I'm repeating the steps with the S. epidermidis. I am going to begin exercise 2, titled Biochemical Testing for Microbial Identification, the catalase testing. I have my E. coli and my epidermidis plates, and I have my slides prepared. So I'm going to swab the E. coli with an inoculation loop and apply it to the slide.
1 drop.
I've applied the E. coli to the slide, and I will drop 1 drop of hydrogen peroxide to the slide.
We have
And it is bubbling. And I'll continue with the S. epidermidis using the inoculation loop and applying to the slide. Oh, that smells bad. And 1 drop of hydrogen peroxide. I don't see any bubbles. The 2 vials of MRVP have been incubating for 48 hours. I've taken them and applied half of the E. coli to each test tube and half of the S. epidermidis to each test tube. I'm going to apply 7 drops of the methyl red to 1 of each of the tubes. Now I'm applying 12 drops of the Barrett's A into each test tube.
No.
Now I'm applying 4 drops of the Barrett's Beast solution. I'll allow it to sit for